Hi everyone, this is the last part from the chapter on biological molecules, okay, part 5. Now in this video, right, I'll be talking about the practical component from this chapter, okay. In the past 4 videos, right, I'm just talking about the theory on the nutrients, and for this particular chapter, right, there is a very unique practical component that is able to test for the presence of some nutrients that is from the O-level syllabus. Okay, so we call this food test. And so let's begin. All right, from the syllabus, right, you have to describe. Okay, you can also answer this in theory and practical. Okay, you can describe and carry out tests for 4 particular nutrients. All right, you can test for the presence of starch using iodine solution. All right, you can also test for the presence of reducing sugar using this very unique reagent known as Benedict's solution. You can test whether a food sample, right, contain protein or not using a reagent known as burette solution. And finally, right, you can test whether there is presence or absence of fats using ethanol as a reagent. Okay, so not only do you need to learn how to practice them in practical, you must also learn how to describe the test in a theory question. Okay, that's the reason why I have created a video for this. Okay, because it is still pretty heavy in theory. Okay, so for food test, right, you can test for these 4 nutrients. Okay, starch, reducing sugar, fats, and proteins. You can test for these 4. Okay, when you test for each of them, right, you have to ask yourself 4 questions. Okay, number 1, what is the reagent used for the particular test? Number 2, how do I describe the procedure? Okay, what is the procedure? What is step 1? What is step 2, step 3, 4, and onwards? Okay, what is the observation that I should be looking at? Okay, if I see this coloration, what does it mean? If I see another coloration, what does it mean? Okay, so you must link the observation, right, to the conclusion, all right, which is of course for the presence and absence of the nutrient. So these are the 4 major questions that you need to know. Okay, regardless whether you are doing it as a practical or in theory. So let's start with starch, all right, or what we call iodine test. Okay, the reagent, right, is known as iodine solution, and I think many of you are familiar with this because of what you did in primary school, I presume. Okay, so the reagent for starch test, right, is iodine solution. The procedure, right, is very simple. Okay, you add 2 to 3 drops of iodine solution on the food sample. All right, and it is just like that. It is a very straightforward procedure. What you will observe and what can be concluded from your observation, right? There are 2. Okay, if the iodine solution, which is already yellowish brown, and it chooses to remain as yellowish brown on the food sample, it means that there is an absence of starch. There is no starch. Okay. However, if there is starch, right? If there is starch, you can see that there is a color change. Okay, the color will change from yellowish brown to blue black. And if you see this change in coloration, right, it means that the food sample contains starch. All right, so this is starch test, very straightforward. The second test, right, to remember is the test for reducing sugar. This is also known as Benedict's test. Okay, so if you see the question writing Benedict's test, right, then you should be thinking about this. Okay, the reagent used, right, is known as Benedict's solution. The procedure is firstly, right, you have to extract the nutrients out from food sample by adding water. Okay, so we call this decant. So a food sample is solid, right? You need to extract the nutrients out by adding water in Benedict's test. Okay, uh, after you extract the liquid out, okay, through adding water, we call this a decant. And you don't need to use all the decant. You just need about 2 cubic centimeter. Okay, so you extract 2 cubic centimeter of the liquid into a test tube. Then you add the same amount of Benedict solution to the decant. Okay, so it is of 1:1 proportion. After you add Benedict solution to the decant, okay, you must ensure that the solutions mix well. So what you do, right, whether or not it's practical or theory, you have to shake the test tube of contents, okay? And when it is after you have shaken the test tube, right, to ensure that the solutions mix, okay, you must place the test tube into a boiling water bath for about 5 minutes. Okay, so in this 5 minutes, right, if there is reducing sugar, the solution will change color. Okay, if it doesn't change, then it will remain blue. So the observation, right, is actually tied to the conclusion. Okay, so this is a picture that shows you the change in color. If it's just this color, right, that I'm circling right now, it means that there is no reducing sugar present. Okay, because this is the color of Benedict solution. So if the solution remains blue, right, then there is an absence of reducing sugar. Okay, reducing sugar, by the way, is a type of carbohydrate. Now, if the solution turns from blue to green precipitate, okay, PPT means precipitate, it means that there is small amounts of reducing sugar present, like this, the one that I'm circling right now. Okay, you can see that it is slightly greenish in color, or you can see little traces of powder inside, which we call precipitation. Okay, if you see about this color and it doesn't change anymore, it means that there are small amount of reducing sugar. Sometimes, right, the food sample will continue to change color and it might change to yellow. Okay, it might change to a yellow precipitation. It also means that there is small amount of reducing sugar. Okay, now when it changes to orange, which is over here, this one that I'm circling, okay, if you change from blue to orange precipitate, you can conclude, right, that the amount of reducing sugar in the food sample is of a moderate amount. That means it's a large amount. Okay, and finally, right, it will change from blue to brick red precipitate sometimes. It means that the food sample has a large amount of reducing sugar as well. Okay, so over here, right, these two coloration means it's a large amount, and these two coloration, okay, means it's a small amount. All right, so the color, right, of the observation change can actually tells you the quantity of reducing sugar present in the food sample. All right, so let's move on to the test for fats. Okay, now this is also known as ethanol emulsion test. All right, the reagent used, right, is ethanol followed by water. The procedure, right, goes like this. Okay, so firstly, you add ethanol to the food sample. All right, if the food sample is solid, you add ethanol to it, because ethanol, right, will ensure that if there is fats, right, the fats can dissolve into the ethanol. So this is the reason why, right, for this particular test, you don't add water, you add ethanol first. Because if you add water, the water is not able to extract the fats. Okay, because fats are soluble in alcohol. So we normally use ethanol. All right, so for this particular test, you have to add ethanol so that they can extract the fats. You transfer the decant into a test tube. So after you shake, right, and you shake for a while, and then hopefully there is fats and the fats will be dissolved into the ethanol, you can pour the ethanol into another test tube. Okay, and this is also a decant. Now what you do to this decant right now, right, is that you pour the decant into a test tube containing water. So yes, you have to transfer quite a number of times. All right, so when you pour the decant into a test tube containing water, right, if there is fats, right, you will be able to see changes. Okay, if there is no fats, the solution, right, will remain clear. It means that if you pour the decant into water and it is still transparent, right, it means that there is absence of fats. However, if the solution forms a white emulsion, okay, like this, all right, if the solution forms a white emulsion, it means that fats is actually present in the decant. So a white or white cloudy emulsion, right, means that there is actually fats present. Lastly, let's talk about the fourth test, which is the test for protein. Okay, this is also known as Buret's test. So if you see Buret's test, please do not confuse yourself with Benedict's test. Okay, Benedict's test is for reducing sugar. Buret's test is for protein. Okay, the reagent used is known as Buret solution. It is blue in color and it is pretty similar to Benedict's solution. So please read the labels properly. All right, the procedure is the same. Okay, the decant, I wrote liquid here, but it's actually decant. So after you add water and you extract out the decant, right, you only need about 2 cubic centimeter of the decant. Okay, after you extract the 2 cubic centimeter of decant, you add the same amount, 1:1 proportion of Buret solution to the decant. Okay, then you shake the test tube, and if by now there is no color change, it means that there is no protein. All right. So your observation got two types as well. If the solution remains blue, it means that there is absence of protein. However, if the solution turns from blue to violet, it means that there is presence of protein. Okay, so over here, right, Buret solution is this color, it's already blue. So after you add Buret to the decant, right, if it still remains blue, it means that there is no protein present. However, if you add Buret solution to the decant, okay, and it becomes violet, it means that there is presence of protein. Okay, so the coloration actually tells you whether there is protein or not. All right, so that's it. Okay, this video is just talking about the 4-foot test, which you have to know through practical and also in theory. And of course, in theory, you have to describe the procedure, you have to state the reagents, and depending on the context of the question, you have to identify the positive outcome or the negative outcome. Okay, and link it to the conclusion. All right, that's it. Thank you. Bye bye.